Hello, everyone. Very welcome to this session. We will discuss today with two excellent friends and colleagues about the Presidential Symposium of today. My name is Alessandra Curioni. I'm from the University Hospital in Zurich, and I'm the editor-in-chief of the Daily Reporter. Today with me, there is Antonio Passaro from Milan, from Italy, and I see there is already Juan Valle, who I'm going to ask directly, immediately. Juan, it has been for you certainly a very important day today with this data of the trial of the first MAP. How can you bring this, please, this rare disease into context, and how does this change your practice?
Yeah, thank you very much, Alessandra. This is a very exciting day because we've really been in an evidence-free zone in patients with metastatic pheochromocytomas and paragangliomas. The issue here is that these are very rare malignancies, and we're talking about the incidence of 1 in a million. So it's always going to be very challenging to undertake prospective clinical trials. And I'd like to congratulate the international team led by Eric Baudin, who evaluated sunitinib. And this wasn't a guesswork. This was, based on some preclinical data, some scientific rationale, and it took 10 years for this study to recruit. Recruited approximately 10 patients per year. Other treatments that we're currently using and are in guidelines, like MIBG therapy and lutetium, don't have the same evidence base. So this is exciting because it's given us a very clear steer of the activity of sunitinib in these patients. What they were able to show is that sunitinib met the threshold for activity to be deemed active. Although it was a randomized study, it was not powered for comparison between the two treatment arms. The control arm was really there to make sure that we could test the statistical assumptions that were being made, and sunitinib then did reach the threshold of activity. So this means that going forward, this is a new treatment option for our patients, one that is safe, particularly with a concern that the malignancy itself can cause hypertension. Sunitinib as a therapy can cause hypertension, but in fact, the collaborators were able to demonstrate that that was safe and manageable.
I think this is, for me, the impressive data. It was exactly what you said for a rare disease to build over the years and the many years such a trial. So at the end, we know that rare diseases, they are there and we have these patients. So I think that this is certainly one of the most important point of it. It was complicated a bit to understand the threshold of positivity, I have to say, because of the confidence of interval. But for you, is this practice changing now? Is now sunitinib for you a treatment?
Yes, it is. And it is for two reasons. It makes sense. It follows a scientific rationale. We know these are vascular tumors. We know that VEGF is overexpressed, as well as some of the other vascular pathways. We see when we're doing multidisciplinary team meetings or tumor boards, these are very vascular tumors. So using an antivascular agent makes a lot of sense.
Absolutely, absolutely.
Yeah.
I think it's very important to see here the statement, probably guidelines is going to change according. And now, is it practice changing? I'm moving to Antonio, completely different field. We are in the field of lung cancer resectable. Antonio, the Empowered 010, is this practice changing for you? And if yes, for whom?
Thank you, Alessandra. So the Empowered 010 is, of course, the first in class for the use of immune checkpoint inhibitor for resecting non-small cell lung cancer. But the results confirm that one size do not fit all, and this kind of treatment maybe is not the best for all comers. The presentation of Enriqueta Felipe was focusing on the site overlaps that did not show any difference in favor of the atezolizumab arm, but confirmed that there is no difference also for the CNS metastasis, that one of the key points for the treatment of lung cancer. But the results confirm that we have maybe a new standard of treatment for patient resected for stage 2, 3A, and in particular for patients with high PD-L1, more than 50%. At the present time, the data for patients with less than 50% with the stage 1B are not so robust to understand the real power of the adjuvant treatment at the present time, and in my opinion, confirm that now there is no indication for these patients. I think that it's time to understand the kind of regulatory approval in this trial, of course, in the US by FDA, but hoping early by also the EMA. And we can suggest to use this kind of treatment from patients with high PD-L1, but we have to wait the more robust data and the approval requirement.
So your high PD-L1, you would say now from stage 2 to 3 with PD-L1 more than 50% or more than 1%? What do you expect is going to be the approval?
But the data, considering the data, we were only to wait the approval for more than 50%. So I understand that this is a secondary endpoint, of course, but the other ratio for disease-free survival for patients with 1/49 or less than 1 are not so positive to approve this drug in this setting at the present time. Of course, this is in adjuvant setting, and we have to wait also for more robust data about the overall survival, but for less than 50%, these data are not clearly for an approval.
Like all adjuvant studies, disease-free survival used as a primary endpoint. We are waiting for the overall survival. We are also waiting for the data on patients that have molecular alterations with oncogenic driver, right? Today we have seen some data with EGFR and ALK. And so probably this population of patients, we always ask to ourselves, should we give or not immunotherapy? So there are still several data in that sense to come. And are you doing it just very quickly, molecular testing now for your patients in the light of the ADAURA trial and this Empower 010? Are you doing molecular testing?
Yes, this is a clear clinical need at the present time, particularly for patients with EGFR positive disease. ADAURA showed a great improvement, a dramatic improvement for our patient, and testing our patient for EGFR now is recommended also in clinical practice.
That's kind of very important. And about testing and monitoring patients, let's try very briefly to say the impact of the data that we have seen today with the CAPTURE trial for about patients with COVID and cancer and the immune responses. A wonderful study because finally we have data on the cellular response, right, in such a broad analysis. And then afterwards, the other study that was showing the antibody responses specifically in patients on treatment. Do you check and the question goes to both, so maybe first of all to Juan, do you check the antibody titer in your patients who are under treatment with cancer, solid tumors in general?
No, yes. It's not our routine in clinical practice to check the antibody.
So, Antonio, you don't check it. Juan, do you check it?
No, at the present moment, it isn't something that we check routinely. I think in order for us to do so, we would really need to have a very specific purpose for doing so. I think these are provocative results and may well lead to some additional thoughts about testing in the future.
Yes, because probably the major question is if we would have a patient that after the second vaccination doesn't have any antibody to be detected, should you give the third? Do you give it in UK? The third?
Not at the moment. There's just about to be a rollout program for the third vaccination, but that is being rolled out for all vulnerable patients, which will include patients receiving chemotherapy, but it will not depend on antibody titers.
Which is actually very important because it's what we have seen today, how important it is after the second vaccine, right? We have seen this already from previous study, but today I think it has been, the impact on the cellular responses has been really amazing. Antonio, yes or no? Do you do the third vaccine for your patients at the moment in Italy?
Hoping yes. Not today, but hoping yes.
I think we will also start very soon. So this was a very quick wrap-up. And of course, stay tuned with ESMO 2021. This has been a real pleasure for me to have Juan Valle from UK and Antonio Passaro from Italy. I think it has been a very intensive day today, also the Presidential Symposium. You can look at it also later on the website. It's a great pleasure to have had you online today with us, and we'll see each other tomorrow. Have a wonderful day and a great ESMO 2021. Thank you, everybody.